Okay, so now what type of, like, as I was mentioning, the types of bond that could be in the tertiary structure of protein. So types of bonds in the third degree or the tertiary structure. So one of them could be van der Waals, as I mentioned. So van der Waals, or you can say instantaneous dipole or induced dipole interaction. So instantaneous dipole induced dipole forces between non-polar side chains. I would argue to go back to the AS contents where, like in the bond chapter, you will see all this type of bonds explained in detail. So it might be it might happen that you can't recall certain portion of the bonds, like what type of bond is this, for example, instantaneous dipole, induced dipole interaction. Like you might not be able to recall. It's completely fine. Just go back to the AS contents, like revise or take a look on the the bond chapter, and inshallah you'll be good to go. Then there are other bonds, could be hydrogen bond, as I mentioned. So hydrogen bonding also could be there. Like this is actually in between the side chains, okay, not the peptide chain, peptide region. Peptide region is only in secondary, but in tertiary, the side chain is considered. Then another one could be ionic bond. That means they reacted completely, forming ions. They have no interaction between them as ion, like ionic bond. So this is much more severe, ionic bond. And finally, we have, let's say, covalent disulfide. Remember, there was this another R group, which was the SN group was present as well. Let me see. Yeah, so there was this CH2, sorry, cysteine, SH, yeah, SH. So cysteine was there, okay. So there was the sulfide group, okay. The sulfur was present there. So this is covalent. There is some also stronger bonds. So covalent disulfide, that means between two sulfide group, that means one from each peptide linkage. So let's say there's one chain as another chain, and both have sulfide. So between that sulfide, this bond could be formed. That bond, like they're talking about. So covalent disulfide. Di means two, and two means why? Like why two in this case? Because both the chain needs to have what? Sulfide. So disulfide bridges, covalent disulfide bridges formed between cysteine. So for this bond, specifically we need what? A cysteine molecule. I'm sorry for that. I think things are much more fine now. So as I was mentioning, that for this sort of reaction, we need what? We need like simply cysteine is needed. Cysteine residues are needed. Cysteine residues are needed for such reaction. Okay, so at a different location in the primary sequence. So that means in the sequence of the, let's say, the primary sequence means what? Now let's define the primary sequence. Primary sequence means just normal single chain. There's no other interaction, no hydrogen bond, no like the side chain bond. So nothing was there. Just simply the peptide bonds were there. That's known as the primary structure, where it said primary structure is the sequence of amino acids in a polypeptide chain. So that means simply just the monomers are bonded with each other, forming that peptide linkage. Okay, I hope that's much more simpler to understand now. Now, let's see some type of like examples. What type of like residues are present there? So let's say residue one. Now residues are like what? Residues are the, let's say, the remaining portion of the repeating unit, you could say. Okay, so let's say we have a large chain. From there, what do we have in common? This peptide linkage part, isn't it? This, let's say, NH, CH2, CRH, and CO group, isn't it? This is the repeating unit. It repeats itself. So this is known as the residue. Okay, like if we break it into simplest form, what is the residue we are getting? For example, when we filter something, we get the residue, isn't it? We get in a filter paper, we get the residue. Similarly, that's also the same thing. We get like if we break them, if we break the polymer into the tiniest particle, the residue is the simplest repeating unit possible. So there's a residue one, let's say, and we have residue two. And we are also saying in between residue and residue two, what type of tertiary bonding could be possible? Type of tertiary bonding. Okay. So let's say what type of residues could be present. Let's say one of the residue is, let's say, HNCH. Wait a minute. Yeah. So HCH, then there's this, maybe somewhere like previously I have mentioned something wrong there. There was like CH, not CH2, rather CHR directly and COO. This was the case, isn't it? Yeah. So I most probably mentioned something wrong there. So this is the actual amino acid structure. Let me see. Where did I mention this wrong thing? So here, this shouldn't be there. So I'm sorry for that. This is the structure. Like eliminating this part, rest of the part is the amino acids part. Okay, yeah. Now from there, from here we get the, let's say, this is our side chain, which is CH2, 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 and NH2. Finally, we have CO group. This is the residue one. The residue two is, let's say, H, N, CH, CH2, CH2, COOH, and we have CO group here. Then we have, let's say, H, N, CH, CH3, CO. Remember, you have to remember which portion is which one. What is the name of this molecule? What is the name of this molecule? Like you have to like remember this portion. Okay, that's the only difficult part of, I would say, mostly in the organics part. So since like in biology, you have also most probably have read these parts, but even if you didn't, you have to like know. But you'll like eventually with practice, you will know which molecules are which. For example, and this is, I also can't recall now right now, but in my exam, I had to like remember what was this. Like, so this is actually glycine. No, sorry, this is not glycine. This is alanine. I'm really sorry. This is alanine. Then this is known as lysine. This is known as aspartic acid. Yes, aspartic acid. So yes, you will eventually get to know what are these molecules, inshallah. So here we have HNCH, CH3. That means another glycine. So glycine bonding with glycine, what sort of reaction will be there? Remember, this is, we are talking about just tertiary. So HNCH, CH2SH. So all the possible type of bonds will be shown here. So you have to just identify them. I would urge you to pause the video after I've drawn all the structures and try to yourself by seeing the types of bond from the tertiary part, like the notes just given over here, the types of bonds in the tertiary structure. See the type of bond, only 4 types of bonds are there, and identify, distinguish which bond or which residue will form which bond. And we have HN, CH, CH2, OH, CO. Finally, the other residue is H and CH, CH2, COOH, finally CA. Okay, yeah. Now let's see. So pause the video and try it yourself. This is the last part of today's lecture. So here we have what? We have amine with carboxylic acid. That means what? There will be an what? Ionic bond, isn't it? So here, remember, more than 2 types of bond could also be formed. Okay, not necessarily only one bond will form. Here, in this case, ionic interaction, or ionic attraction, you can say attraction, ionic attraction, as well as what? The carbon has oxygen, nitrogen has hydrogen, what? Hydrogen bond, H bonding. Okay. Over here, we have what? CH3, CH3, that means only van der Waals. Okay. Then we have what here? We have sulfide. Both are cysteine. That means this is disulfide bond. Disulfide bond. Finally, we have what? Carboxylic acid and alcohol here. That means what? Here will be hydrogen bond. H bonding. Okay. Carboxylic acid and alcohol does not like undergo ionic reaction. Why? Because like simply think like what sort of reaction happens here? Here basically the reaction happens is what? Acidic, like this is an acid and this is the base. So acid base neutralization reaction happens here. And when neutralization reaction happens, what happens? COO minus and what? NH3 plus, isn't it? And they tend to form like ions and bonds. So carboxylic acid loses H plus and amine gains H plus. This is the structure formed, and they have interaction between them. That's the ionic bonding. At the same time, this nitrogen or this carbon has this C double bond O. Okay, and that's the O minus, and N, let's say N has this hydrogen here, and the other H's are, let's say this is H plus here, and this is another H. So this NH3 total, and there's the carbon like bonded with the other part of the carbon. What happens? These hydrogen and oxygen, they tend to form what? Hydrogen bond. So yeah, that's how it works. Try to draw structures and then imagine, okay, what I'm trying to say. Try to draw them in their structure, okay? Or try to draw the discrete structure. That will be much more convenient for you. Okay. Now, just a short description on what we'll be learning in the next lecture is the bonding types. So bonding type as well as structure involved. So we have, let's say the bonding involved, let's say disulfide, okay? Disulfide bond, okay? Between chain. Okay. Then we have, let's say, H bond in beta plated sheet. Then we have, let's say, ionic bond between parts of chain. Parts of chain means the like the polypeptides, the chains, like certain part have ionic bond in between them. And so this is the ionic bond. This portion forms an ionic bond with another portion of the like the chain, the polypeptide chain, okay, the polymer. Then the fourth one is peptide links between amino acids. Now you have to identify which one is which type of like structure is involved. I would urge you to pause the video and try it by yourself. This is the homework for you. Okay. No need to actually pause the video. I will end here. So try it yourself. We will inshallah be seeing the solution in the next class. Okay, take care for today. Assalamu alaikum.